Okay. So I'd like to start this talk off by again thanking the competition organizers. It's a great honor to be here. I'd also like to make a confession. I have never studied bacteria in my entire life. I'm actually a computational neuroscientist who's interested in building models of neuron dynamics, and the problem of parameter fitting comes up in that context quite a lot. So I started playing around with this competition outside of my day-to-day research, and I was quite surprised to be invited here. So I hope to maybe bring a little bit of my prior knowledge to bear and contextualize this competition in the broader field of computational science, in addition to talking about the specifics of this competition. Okay, so for the first half of the talk, I'm going to summarize what I actually did in this competition, and I'm going to go through most of the important details, but I am going to gloss over a few things because I want to push through and get to some high-level questions that I think have already sort of been touched upon by the previous presentation and the introduction. And the first problem is that disparate parameter sets in this bacteria model can produce similar phenotypes. And this is why, even if you have a really good parameter distance, as we've been calling it, you don't necessarily have any better prediction distance. And in my lab at Brandeis University, we call this the problem of multiple solutions. That is that you can construct disparate biophysical models that recapitulate biological data equally well. And this is a problem for parameter fitting because if you want to find the quote-unquote true parameters of a biological system, you have no way of choosing between parameter sets that give you equally good performance. So I'm going to give a tentative suggestion or solution to this problem, which is to use rich and complex perturbations to unmask hidden differences between superficially similar biological models. And then at the end of the talk, if I don't run out of time, I want to touch on some more technical details of dimensionality reduction. As part of my method, I used principal component analysis out of necessity. And I think to really make more progress on this problem, we need to start looking towards contemporary nonlinear dimensionality reduction algorithms, which is really a hot topic in the machine learning community today and is really an unsolved problem. Okay, but let's start off with what I actually did. When I first started playing around with this competition, my gut instinct was to use a genetic algorithm to optimize the parameters of the model and minimize prediction distance. However, I quickly realized that I didn't have enough personal computing resources at my disposal to do this. As has already been mentioned, it takes about 24 hours to run a single iteration of this model. And similar observations were made by other teams on the community forum. So I landed on a different approach using statistical inference, in which I tried to run as many parameter sets as I could just to get an idea of how the model operated. So I would pick a parameter set and simulate that model, which takes about 24 hours, and record the phenotype of that model. And then I would choose a different parameter set, record its phenotype, and repeat this as many times as I could. And then at the end of the day, the competition organizers gave us a target phenotype we were trying to reproduce. So the clear way forward is to train a statistical model or machine learning algorithm to predict the parameter set from the phenotype, and then use that model to then go from the target phenotype to the target parameter set. So the simplest statistical model you could develop, or you could imagine developing, is a linear regression algorithm that takes a number of independent variables x and looks for a linear relationship between those variables and dependent variables. This, although I did, just for the sake of it, explore this pathway, fails miserably because this model is highly nonlinear, as has already been covered. So a second way forward, which I didn't end up taking, but it would be an interesting future direction, would be to use an artificial neural network where you have a layered architecture and your independent variables are put in on the input layer, and your dependent variables get spit out the other side. This has been shown to approximate any arbitrary nonlinear relationship, so that's what's nice about it. I ended up using a different approach, which is very similar in spirit and has actually already been mentioned today several times, which is the random forest algorithm. For those of you who aren't familiar with this, all you really need to know for the sake of this talk is that it's a very powerful technique that is able to approximate arbitrary nonlinear relationships between a set of independent variables and a set of dependent variables. The way it does it is it uses the predictor variables in the training data set to stochastically develop many regression trees. And because these regression trees are stochastic, each one of them has some idea of what the dependent variable is, and they each get a vote. And you basically pool your estimation across all these distributed regression trees, and that is your predicted response. Okay, so let's be concrete about this actual competition. So I simulated as many phenotypes as I could, and I consolidated all the data on those phenotypes into a giant matrix, which I'm calling X. And the width of this matrix is the number of cell variables that the code spits out and characterizes its phenotype. And in my implementation, this was roughly 130,000 independent variables. And because the model takes so long to run, and because I had limited computational resources at my local institution, I was only able to run a total of roughly 1,100 simulations. So this is a very poorly conditioned data set. I have many more independent variables than I do replicates. And just so everybody's on the same page, the way this matrix is organized, the first column might be the concentration of a certain protein across all simulations I did. The second column would be a concentration of a different protein or some other cell variable. The first row would be the first simulation I ran, and the second row would be the second, etc. So in order to make this tractable statistical fitting algorithm, I needed to do dimensionality reduction to reduce this large matrix into something more reasonable. And I ended up using principal component analysis because that is the sort of classical and most well-established dimensionality reduction technique. And it's actually been shown to outperform many proposed nonlinear dimensionality reduction techniques. So this was a good place to start. However, I do think in the future this approach is insufficient, and it will be interesting to see over the next 5-10 years as more nonlinear dimensionality reduction techniques are introduced, whether those can improve performance. But the basic idea is now I have a reduced set or a reduced description of each cell's phenotype in this matrix X prime, and I match that with all the parameters that generated those phenotypes in a matrix I'm calling Y. And then I did something a little silly, but I think this is the correct way to do it. Rather than predicting all of the parameters in one fell swoop, I decided to use an iterative approach to fit the parameters one at a time, and that way I could incorporate as much possible information into the model as possible. So here's how this went. I first randomly selected a column of this Y matrix to predict, so one of the 30 possible parameters that the competition organizers identified. And then I used the target model phenotype, again in reduced principal component space, to predict this particular parameter which I had randomly selected. Then I chose I randomly chose one of the remaining parameters to fit, and I incorporated the value of the original parameter that I had already fit. So just to be clear, this would be the first iteration where I fit one parameter, and then in the second iteration, I choose a new parameter to fit, and I train the random forest algorithm to fit that parameter based not only on the target phenotype, but on what I had already predicted for the first parameter. And I would continue this for 30 iterations until I had predicted all parameters. So that's what I did on a high level. Here are some concrete results. So this plot shows the principal component analysis. And the y-axis is showing the cumulative percent unexplained variance. And you can see that there's a very heavy tail in this distribution, which means that you need to consider many principal components to capture all the variability in the training data set that I generated. So this is somewhat discouraging, but as has already been explained, this model is highly stochastic. So part of my hope was that a lot of the variance in this heavy tail reflected noise that would not be useful for the random forest model to consider in the first place. So I took the top 50 components, and really I tried several things. I tried using the top 10 components, top 100 components, even top 500 components at one point. And I didn't see too much difference in the performance of the algorithm. It just took shorter or longer. But one thing that I do want to note is that if you look at the top 50 principal components, you're still capturing roughly half of the total variability across the entire data set. So this is at least, it was somewhat encouraging to me when I first looked at this. Okay, so this is a plot showing how close the algorithm actually got in terms of the parameter distance, as has been mentioned already. So here, the y-axis shows the value of each of the 30 parameters, which are just listed in a somewhat arbitrary order. The black dashed line represents no change in the parameter from the wild type model that is open source. The blue circles is the final solution that Jonathan Carr gave me after the competition was over through personal correspondence. And the red X's show different estimates that the random forest algorithm I just described to you produced. So you can see in the parameter distance, I really didn't get all that close. And you can also see that just because of the stochasticity of the method I used, there's already some variability on what the actual estimate is. So I got this set of estimates, and I also performed just a random local search in this area to see if I could do better. Okay, so this leads me to the problem of multiple solutions. So just going back to this for a second, this is showing that I wasn't able to find the parameters directly. However, as has been mentioned, the prediction distance, or the phenotype of this and other models I simulated, actually didn't do too bad. So I was able to reproduce the phenotype without getting the exact parameters. And here's an example that can show you how this is the case. Here on the right are two parameter sets on two different models, or two different parameter sets that I created randomly. And on the left is a plot showing the phenotype of these two parameter sets. So you can see that the parameters of these two models are incredibly different, but if you look at the top 50 principal components, they're almost identical. And the black dashed lines here are showing the 95th and 5th percentiles for each component. And the y-axis is showing the magnitude, or the score for that component. And again, the principal components are ranked on the total amount of variance that they're explaining across the whole population. So that might be a little abstract, so let's look at some single cell data, or growth curves. So remember that the competition organizers created a mutant model that grew much slower than the wild type model. So the blue lines are showing growth curves for the target phenotype that the model or the competition organizers gave us. And remember, because of the stochasticity, there's quite a bit of variability here. So that's what's in blue. In red is an example parameter set. I don't even think it was the best one. That shows a single growth curve of a simulation I ran that is essentially well fit to the average of this population of mutant models. So this is again showing that even though I wasn't able to get the parameters, certain aspects of the cell's phenotype were recoverable. And now I want to change gears and talk about similar observations in the field of computational neuroscience, a lot of which has come out of my lab, the lab that I work in at Brandeis University. These are 4 papers here. The top 2 are theory papers that deal with this problem in computational models of neuron function, and the bottom 2 are actually experimental papers that show that even real biological cells, even though even when they show similar phenotypes, actually can achieve those phenotypes by having diverse biochemical parameters. So the problem of multiple solutions is not just one for theorists, but one for experimentalists as well. And I won't have enough time to really go into that. But let's talk about some computational models of neuron function. Here, this is showing the output of a model. It's creating a set of bursts of action potentials or electrical discharges. And these bars are going to represent the parameters that produce this model output. If you change these parameters by plus or minus 20%, you can find many different phenotypes for this neuron. Many different bursting and just tonically firing patterns to neurons that are completely silent. However, if you change and modify the parameters over an even larger range, plus or minus 50%, you can still find many different parameter sets that give you similar output or phenotypes. So here, we would call this a one spike burster phenotype. And one interesting possibility to overcome this problem of multiple solutions is to add a rich and complex input to the system that perturbs output. So here on the left are two computational models of a bursting neuron. And in this case, they're given no input. On the left here are the same two models, but in the presence of a pink noise stimulus, where current is injected directly into the model cell. So this on the left could be the basal activity of the cell if it's left unperturbed. And on the right is when we experimentally perturb it. And in this competition, in this dream competition, we were working with a bacteria cell that was largely under these conditions. So it would be interesting to apply this methodology. So how would we do this? Let's say we have two models of a bacteria cell that produce relatively similar phenotypes. I'm showing here average growth curves because that's pretty easy to interpret. So it's difficult to distinguish which of these models is correct because they're producing very similar outputs. But now let's say halfway through or after some time into the simulation, we add a perturbation, such as the addition of a chemically or biochemically active substance. Perhaps this perturbs the growth curves for one of the bacteria cells, but leaves the other one slightly unchanged. And now there's a slight difference between the two growth curves of the two cells. This would help somewhat, but maybe the phenotypic variability is still not enough. What the optimal solution might be is to use a multifactorial approach where you deliver many different chemical substances at different times in the simulation, and this might cause an even greater divergence of the growth curves. So it would be interesting to utilize this technique, and this might help, especially if rather than fitting 30 parameters, we want to fit all the parameters in the model, which I believe is over 1,000 at least. So the last problem that I want to talk about is dimensionality reduction, and this is, again, a little more technical. But I've only used the top 50 principal components in my algorithm, and after going back to the data, I believe that this is a shortcoming of the technique that I'm proposing here, and it's really a limitation on the principal component analysis itself. So here is the target phenotype in blue, and X is the closest simulation that I submitted showing the top 50 principal components. So you can see that the phenotype, or this description of the phenotype in principal component space, isn't perfect, but it's all right. But if you consider not just the top 50 principal components, but all principal components, and you look again at the target phenotype, you see this very interesting cluster of principal components, all of which have a high score or high magnitude, suggesting that there's really something important going on out here. And just to put this in perspective, if you look across the entire population, or the training set that I simulated, the principal component analysis told me that by the time you get up to component 400, roughly 80% of all the variance is explained in the population. So even though I'm explaining a lot of variance at the population level by considering the top 400 components, let's say, I'm still missing a very key aspect of this cell's phenotype. This is a blow-up of that section showing the target solution in blue and the closest simulation that I had for components 400 to 425. So in all 1,100 simulations I ran, I never found anything that really replicated this, this interesting pattern here. So what are the take-home messages? The first is that the most important principal components, as you would define them from a population level, don't always capture these idiosyncrasies of individual phenotypes and are not always the most important components if you're considering an individual. And the second observation is that nearby components appear to be correlated in complex ways, so that in the target phenotype there is a burst or a cluster of significant components around 400 to 425, but not after that or before that. So that to me suggests that this space is highly nonlinear, and the linear principal component analysis is having a hard time fitting these nonlinearities, so it has to do it in over 25 components. And this again suggests that we could do better by looking at nonlinear dimensionality reduction techniques. So this is my last slide, what needs to be done. As I just said, nonlinear dimensionality reduction is a really interesting approach that I'm interested in looking into in the future. Unfortunately, I'm running out of time, so I'm not going to go into the details of what I think would work best. The second point that I've told you about is the problem of multiple solutions and the ability to unmask latent variability in superficially similar models by using rich and complex perturbations. And finally, more than anything, one of the biggest limitations in this dream challenge was the amount of computational power available to me on my local cluster. So it would be interesting if in the future this dream challenge not only provided cloud, the ability for participants to simulate the model on the cloud, but actually gave participants pre-simulated data sets and turned this into more of a data mining exercise than an exercise in who has the best cluster or fastest processor. So with that, I'd like to close quickly. Unfortunately, I don't have enough time to really go into detail here, but I'd really like to thank the principal investigator of my lab, Eve Martyr, and everybody in the Martyr lab for all their support. I really wouldn't be here without them. I'd also like to give a special thanks to Jonathan Carr, who really went above and beyond what any organizer for this competition would need to do and helped me debug code and work out bitmill issues. Dr. Ted Brookings in the Martyr Lab contributed the slide with pink noise to this presentation. And finally, my teammate, Jeremy Zucker, who lives in Jamaica Plain. And thanks again for listening.